central brains of the Drosophila. Okay, so I've developed a series of specific research questions that I want to ask, and I've been spending the past year trying to answer these questions. So the first one I'm asking is, are Wolbachia localizing to neurons? So we know that Wolbachia are found within the central brain of the Drosophila, but it hasn't been observed if they're actually localizing to the neurons, so that's the most important thing we want to find out. And if we find out that they are localizing to neurons, then secondly, we want to know what part of the neuron do Wolbachia localize to. So I have pictured here just the basic structure of a neuron, and we want to know if Wolbachia are present in the cell body, which is where the host DNA is contained within the nucleus, or if Wolbachia are localizing to the axon. And this axon is involved in signaling within the brain. Also, we want to analyze if Wolbachia localize to specific groups of neurons. So the Drosophila central brain is divided into these specific regions, and all of these regions are just composed of groups of neurons. So we want to know where the Wolbachia are localizing, which of these regions they're localizing to. And lastly, we want to know if Wolbachia can live extracellularly outside of the neurons. So if it's possible that they're occupying the space around the neurons. Okay, so a lot of my research was actually done at the University of California, Santa Cruz. And I was able to use FIRSCA funding to travel to this lab and to work in their lab for a while. And basically, each experiment that I performed had the same basic principle. And the main goal was to visualize the Wolbachia DNA within the host brain. So in order to visualize these brains, first I had to dissect a lot of adult Drosophila brains, which is pretty tedious work. And then I stained them with antibodies. And after doing this, I took images of the brains using a confocal microscope, which is pictured at the left. And this antibody staining is what labels the brain these different colors, in this case green and red. And this is important because it allows us to differentiate between the brain structures and between the different DNA found in the host brain. Okay, so first we wanted to find out if Wolbachia are localizing to neurons. So here's a picture of adult Drosophila neurons, and labeled in green is the cell cortex. So this is the outside of the host cell. Labeled in blue is host DNA, and labeled in red is Wolbachia DNA. And it's really easy to see that the Wolbachia are localizing to these neurons. So the Wolbachia are contained within the cell cortex. So with this data, we were able to determine that yes, Wolbachia do localize to neurons within the central nervous system. Okay, well great. What part of the neuron are they localizing to? Once again, we're trying to find out if they're localizing to the cell body or to the axons. So this figure is a close-up of the Drosophila central brain, and pictured in green are axons, and pictured in red are cell bodies. And what we found is that there were these large aggregates of Wolbachia that you can see here, and these aggregates were only found in the cell bodies. They're not found in the axons. So as you can see, there are none of these axons, which allows us to determine that Wolbachia are localizing to cell bodies, but not to these axons. So we also wanted to analyze, do Wolbachia localize to specific groups of neurons? And the first region that we looked at were mushroom bodies. And these mushroom body neurons are this structure right here. It kind of looks like a goalpost. And the reason we chose this region is because mushroom body neurons are involved with learning and memory within the host, as well as olfactory senses. So this is a really important region to look at. Okay, so we did an antibody stain for these neurons. Once again, this is a nice picture of the structure up here. And we found that Wolbachia were not present within these axons. And this is actually consistent with our other data that Wolbachia weren't present in the axons. So once again, pictured in green are the axons. So this allowed us to determine that they aren't present within these axons. However, this doesn't mean that Wolbachia aren't present in the cell bodies. Just right now, there's not an antibody that's specific to these neurons. So once we kind of further develop this experiment, we're going to continue to test if Wolbachia is present within this important region of the brain. Okay, so lastly, we wanted to determine if Wolbachia could live extracellularly outside of the neurons. And as we were taking images of these brains, we found that there were these large aggregates of Wolbachia. I mean, these are just huge masses of bacteria, pictured here as well as these areas. So this led us to believe that the bacteria were not contained within the cell cortex. There's no way that this is, a normal cell and that these are within one of the host neurons. Okay, so this data is suggesting that it is possible that Wolbachia can occupy the extracellular space. And this is actually a really surprising finding because this hasn't been observed in the Drosophila central brain before. Okay, so in my future research, of course, with scientific research, you're always trying to answer more questions. So I'm really excited to get started on answering these questions. The first of which is, how do Wolbachia localize to the brain? So we know that Wolbachia are found within the Drosophila brain, but we want to know how they get there. One possibility is that they're using the host's circulatory system, but basically, how are they establishing residence within the host brain? I also want to test if Wolbachia are killing host neurons. So as I mentioned, we are seeing these really large aggregates of Wolbachia, and we want to know if essentially they're over-replicating within the host cell, possibly lysing the cell open, which causes the Wolbachia to enter the extracellular space. So we want to know if the bacteria are essentially killing neurons within the host. And lastly, we want to know what the effect of Wolbachia infection is on the Drosophila host. So we know that these flies are infected with bacteria. They have bacterial infection within their brain, and that has to produce some type of consequence, either positive or negative. And we want to figure out what that consequence is. And that's actually where my research is heading at the moment. So as part of my FRISCA project last summer, I was able to present this data at an international conference, and as a result of that, we're now collaborating with a lab in Austria that's looking at the behavior of these flies. And this lab found that altering levels of Wolbachia within the host greatly affects their mating behavior. So Wolbachia within the host can actually have a significant effect on how these flies behave. So we're trying to provide a cellular mechanism for this behavioral response. And this figure is just something we recently put together from the data that was collected from this collaboration. Okay, so overall, just studying the effect that Wolbachia has on Drosophila can, in general, lead to a better understanding of host-pathogen interactions, and we're hoping that the knowledge that we gain from this study can then be applied to other systems and can aid in the research of other host-pathogen interactions. For acknowledgements, definitely a big thanks to my advisor, Dr. Albertson. I'd also like to thank the FRISCA committee for approving my research grant last year as well as this year, and also approving my FRISCA travel grant. A big thank you to the William Sullivan Lab, which is the lab that I worked at in California, as well as Dr. Katerina Lindley, who worked in that lab. And also thank you to our newest collaborator, Dr. Wolfgang Miller at the Medical University of Vienna in Austria. Any questions? Be happy to take questions. Did I explain it that well?
Okay. Is it not to infect the fruit flies with Wolbachia, or you just capture fruit flies when they have the bacteria?
Yeah, surprisingly enough, we just, Dr. Albertson just goes out, backyard and collects wild fruit flies. But they're naturally found infected. There are some fruit flies that aren't infected, but they just get this bacteria within the wild. Yeah, so they're found that way.
If it's not in the mushroom bodies, where do you think it might be, or do you have any other regions of the brain that...
What was that? Sorry.
If it's not in the mushroom bodies, where do you think it might be localized? Do you have any studies further out where it might
Yep,
localize?
I have. We just wanted to look at the mushroom bodies because this is a really important part of the brain. But this is taken from a different paper, and they did find that there were higher concentrations of Wolbachia in certain brain regions. We just wanted to kind of fine-tune that and actually look in axons and look in cell bodies. But there's this lower region, the subesophageal ganglia, and they're found, there's a high prevalence of Wolbachia within that region. Also, the optic lobes, Wolbachia within the optic lobes, but they're just not there at as high of a concentration. Yeah, so they are in other areas. We just wanted to kind of fine-tune that.
That's all the time we have.
Okay. Thank you.